What do we know about the DNA of the Crusaders, and did they leave a lasting genetic mark on the Middle East? Now, the Crusades were a series of religious wars fought between the late 11th and 13th centuries, mainly between Christian kingdoms in Europe and Muslim powers in the Middle East. They were largely driven by the goal of reclaiming Jerusalem and other holy sites, though they were also influenced by politics, wealth and power. Thousands of knights, soldiers and civilians travelled from Europe to the Middle East, leading to the creation of short-lived Crusader states. But what do we know about the DNA of the Crusaders, and did they leave a lasting genetic legacy in the Middle East? Well, the good news is there has actually been proper academic research into this topic. A really fascinating study from 2019 was published in the American Journal of Human Genetics that looked at the genetic impact of the Crusades. And it had some fascinating and surprising findings. Now, at first glance, the answer seems obvious. Large migrations and invasions often reshape the DNA of populations. The Mongol expansion spread direct male lineages across vast parts of Asia, for instance, while European colonisation dramatically reshaped the genetic landscape of the Americas. So surely the Crusades, lasting nearly 200 years, must have left a similar imprint. Well, this 2019 study looked at this question and sequenced the whole genomes of 13 individuals from what is now Lebanon. Now, 9 of these people lived during the medieval period and were from a site in the city of Sidon, a mass burial known as the Crusaders' Pit. This pit contained the remains of at least 25 individuals, mainly showing signs of violent deaths. Archaeological clues, including European-style buckles and a Crusader coin, strongly suggest that these were soldiers killed in battle during the 13th century. They also analysed 4 other people from another site in Lebanon who lived around the Roman period to help them give a representation of the local ancestry before the time of the Crusades. They also compared these 13 samples to databases with thousands of modern people and hundreds of ancient individuals. And when the genomes were analysed, the results were striking. Now, one of the key results is that all the individuals in the Crusader burial were male, but genetically they were far from uniform. First of all, there were individuals who were clearly local Near Easterners, genetically almost identical to both ancient and modern Lebanese populations. Second, there were individuals who were unmistakably European. Some showed strong genetic links to populations like the French, Northern Italians, and even Iberians such as the Basques. One individual clustered closely with Sardinians as well. And then there was the third group, a particularly fascinating one. 2 individuals whose DNA showed a mix of both European and Near Eastern ancestry. This is critical, because it provides direct genetic evidence that Crusaders didn't just fight and settle in the region. They had relationships with the local population, with the local women. And the researchers went further to confirm this. Using advanced genetic modelling, they tested whether these mixed individuals could be explained as descendants of European and Near Eastern parents, and the answer was yes. One individual likely had ancestry combining a Near Easterner, possibly related to Bedouin or Arabian populations, and a European with links to northern Spain or the Basque region. Another individual appeared to be a mix of local Levantine ancestry and a European from a range of possible regions. Now, to understand who these people were better, the researchers then looked at haplogroups, genetic markers that are passed down from father to son in the case of Y-DNA haplogroups, and from mother to children in the case of maternal haplogroups or mitochondrial haplogroups. And if you look at this table, you can see the breakdown of the results, with the mitochondrial and Y-DNA haplogroups on the right-hand side. The study notes that all ancient individuals assigned in this work as either European or Near Eastern had Y-DNA and mitochondrial DNA haplogroups that reflected this ancestry. I.e., Europeans had a lot of the Y-DNA haplogroup R1b and mitochondrial haplogroups H and U5, while Near Easterners had Y-DNA haplogroups like E, T, J and Q, and mitochondrial haplogroups like J1 or HV. However, the admixed individuals SI41 and SI53 had the Y haplogroup R1b-P312 typically found in European males, and mitochondrial DNA haplogroups HV0 and T2 present in both Europe and the Near East. In particular, SI41 carried the DF27 lineage of R1b, which is highly prevalent in Iberia, especially in the Basque region, and this supported their previous results from the autosomal variants that this individual descended from a European related to Northern Spanish or the Basques. The study also found that in the PCA analysis, SI41 overlapped with Neolithic Anatolians, and was distant from any modern West Eurasian population. The combined results suggest that SI41 and SI53 possibly had a European father and a Near Easterner mother, but a more complex admixture scenario where the parents themselves are admixed could also produce this ancestry pattern. So, at least in some cases, European Crusader men are thought to have had children with local Near Eastern women. But what about the lasting genetic impact of the Crusaders in Lebanon? Well, when the researchers compared these ancient genomes to modern populations in Lebanon, they found something surprising. There is almost no detectable genetic trace of Crusader ancestry in modern Lebanese populations. When the researchers sequenced the DNA of people living in Lebanon 2,000 years ago around the Roman period, long before the Crusades, they found that today's Lebanese population is genetically similar to the Roman Lebanese, suggesting the Crusades had no lasting impact on Lebanese genetics. Even Lebanese Christians, who might be expected to show the strongest connection, are genetically very similar to people who lived in the region during the Roman period, more than 400 years before the Crusades began. In other words, despite clear evidence of intermixing at the time, the Crusaders' genetic impact almost completely disappeared. So how is that possible? Well, the answer probably lies in genetic dilution. Even if mixing occurs, its long-term impact depends on scale. If only a relatively small number of people contribute ancestry, their genetic signal can quickly fade over generations, especially when they're absorbed into a much larger local population. And that's exactly what seems to have happened here. The Crusader states were relatively small, and European settlers were vastly outnumbered by the local population. Over time, their genetic contribution became diluted, to the point where it's no longer detectable in genome-wide data. But what does all this tell us? Well, the Crusades were a massive historical event, but likely had a very limited genetic impact. Although this isn't the largest sample size, and it is only one study, the genetic impacts seem very limited. This reminds us that history is often a series of pulses, intense, violent, and transformative moments that can often change the language we speak and the borders of our maps, all without changing the fundamental DNA of the people living there. And speaking of DNA, how do you actually know if you have Viking DNA? Well, to find out, please click here. Thanks for watching, please let me know your thoughts in the comments below, and subscribe and hit the bell, and I'll see you next time.